that we've been working on. I received inquiries from at least an inquiry from at least one person about the posting of the Lecture 1 and the lecture on meiosis. We have got the most of the remainder, at least the terminology portion of Lecture 1 copied, and that will be posted today. The lecture on the orientations, the planes of view, and things like that. I apologize, we don't have that prepared. That should be self-explanatory. We've been implementing that terminology and those ideas already, so hopefully you had a chance to look at those beforehand, and it's coming together for you. The complete lecture on meiosis, I offer apologies again. We just didn't get it together to get that prepared, where everybody's dealing with new technology. We are getting on track, and we will have everything prepared for the future. If you find that the meiosis questions on the exam give you an extremely difficult time, you can inform me of that, and we'll resolve that issue simply by excluding them from the calculation of the percent grade on the exam. It should be review. That's the lecture I actually took straight from my Bio 1 lecture, so it shouldn't be anything that you haven't seen before. So based upon that, I'm comfortable and confident with the approach we're taking. So I appreciate your understanding and your tolerance of this, and we're pretty well geared up now, and we'll push on through. So again, the expectation is that everyone enrolled in the class that is capable of coming into the classroom, we're expecting them to be in the classroom to take the exam on Monday. If for some reason you cannot make it, and again, we'll take it online in the classroom on Monday. If something comes up, you need to inform me through the WebCT mail today or sometime over the weekend. And we will make accommodations and take care of you. In addition, as I have stated, this is an exercise and experience for all of us, and that's great. So this, if everyone does really well, we don't need to worry about it. If it's a complete disaster, we'll come up with a solution to it. But I expect, again, since as you've seen, either the precursor to questions or questions are already embedded within the presentations. You'll see Marco's supplemental lecture on fertilization and the reaction of the oocyte, and there'll be a couple of questions on there for that. It won't devastate your grade. They'll just be questions that you haven't seen before. So I think it'll be a nice balance. If you watch his video, it's only about 12 minutes, I think. That should go fine. So now we are focusing on the formation of the primitive cardiovascular system. And we are working with figures 411 and 412 from the textbook. And we are thinking of angiogenesis because in our mind, we know that we must form, we already have in the back of our mind, in the background of this level of sensibility, that the chorionic villi are developing, the blood lakes are there, the maternal side is exposed, and we need now to begin in the forefront of our sensibilities. We are realizing that the early embryo, the pre-embryo, the embryo needs to establish its circulation to interface with the maternal side through the extraembryonic structures that are differentiating into the chorion and, as we know, the placenta. So within the first week, there's the formation of the blood vessels that will become very obvious. And this takes place in the extraembryonic mesoderm of the yolk sac, the stalk, and the chorion. So in many places all at once. So they form as islands. They're going to expand in size, coalesce, contact each other, fuse together, coalesce, and proceed to form the complete circulation. The yolk in the yolk sac diminishes, so the embryo needs nutrients from another source, and this has been our stance all along. This is intuitive. So this is a lateral view. It's a mid-sagittal section. We see it is at the point where the notochord, the notochordal process is initiated. We know that because the neuroenteric canal, the connection between the amniotic cavity dorsal to the two-layered embryonic disc, is continuous straight through into the yolk sac cavity. So this stages it. We're well into the second week, breaking into the third week, and we recognize the extraembryonic mesoderm surrounding the amnion, the somatic extraembryonic mesoderm, and the extraembryonic mesoderm surrounding the yolk sac, the splanchnic extraembryonic mesoderm. The entoderm is now lining, from the hypoblast, is lining the yolk sac, and the neural ectoderm is just beginning to differentiate as the intraembryonic mesoderm and the notochord would be present. What we are noticing then, as we are breaking into the third week, that in the splanchnic extraembryonic mesoderm around the yolk sac, the blood vessels or blood islands are beginning to form. They will also form in the body stalk and out in the chorion. The primary chorionic villi are quite well established by this time point. Note head end, rostral end, and caudal end. We recall that because we know that the notochord originates from the primitive node. So the primitive streak ends at the primitive node back here. So that's caudal, and this is rostral. And at the rostral end, we know that the heart is going to develop. We know that also in a horizontal plane surrounding this is that pleuropericardial peritoneal canal, pericardial pleuroperitoneal canal. Those extraembryonic coeloms have opened up and formed that horseshoe-shaped space surrounding the organism in the lateral dimensions. And we see that here in this panel B. They're just beginning. Excuse me, they're not showing the extraembryonic coelom here. These are the now the blood vessels that are forming in a similar fashion to the extraembryonic coeloms. So in panel A, what we are seeing is a lateral view of the yolk sac. The amniotic membrane has been cut, and the amniotic disc would be down deep inside here. The connecting stalk has been cut, and we see the wall of the chorion with some chorionic villi. When we look down into this from the this dorsal view, we see the neural plate. We're at the 3-somite stage, and we see in the extraembryonic mesoderm, the blood vessels are beginning to form, and then there are also the blood islands. So there are two sets of structures that are beginning to develop. Blood islands, where we'll have the formation of the blood cells, and then the blood vessels themselves. So we continue now through panels C, D, E, and F to see how actually the blood islands are also forming vessels ultimately. So first there's just a clump of cells. They gather together, start talking to each other, and decide what they're supposed to form. They begin to secrete. Excretion is to the outside environment. Secrete is when it's to the internal environment. They begin to secrete a little bit extracellular or additional extracellular fluid, and they allow that to accumulate nearby. And then as well, some of the cells will become primordial or primitive blood producing cells. Again, we're thinking delineation of the determination, that those final directions that a cell can differentiate into. When we hear these stories, we think of tremendous mitotic activity, tremendous chemical and intercellular signaling. So the buildup of this extracellular fluid creates a cavity, and the primitive blood cells are basically dropped into that fluid, and the blood island ultimately forms a very nice island that has plasma and blood cells in it. And as soon as we begin to fuse all these vessels together and the primordial heart is capable of pumping, we're going to have circulation. So these blood islands form in the mesoderm. They fuse to form the blood vessels. The cells flatten out and form that endothelial, the flat fried egg, sunny side up fried egg type of an appearance of cells that are stretched around the inside of the blood vessels. The blood cells, the hematogenesis, is from the endothelial lining cells. So this is different from adult, but we are forming the very primitive types of blood cells, predominantly red blood cells with hemoglobin in them, so that they're going to carry the oxygen and the carbon dioxide. And the blood vessels in the cells form within the embryonic mesoderm shortly after having formed in the extra-embryonic mesoderm. And so this is a view of what we are wanting to establish, to form these vessels in the yolk sac, in the connecting stalk, and in the chorion, right out into the chorionic villi, excuse me, so that they can be joined with these large vessels, as well as the smaller vessels are forming inside the embryo as well, in the intraembryonic or just the embryonic mesoderm. And then the heart is eventually going to go from in front of the head to be pulled down into the chest, and we form this type of a circulation with the venous drainage through the umbilical vein as we know it into the venous side of the primitive heart, the sinus venosus. Again, we're introducing all of this stuff. We'll do each organ system in detail later. We just want to see what circulatory system we establish. And then as well, there is the systemic venous drainage from the various areas. The yolk sac will be contributing to the gut system. So that's basically the intestinal venous drainage that we're seeing here in the arterial supply. It's through what's called the vitelline artery and vitelline vein into the yolk sac. And then from the arterial side of the heart, the aortic sac will be there, and we feed into arches into the primitive aorta, out through the vitelline artery, and of course all the segmental arteries going to the different parts of the body, or the intersegmental arteries. And we want to feed out through the umbilical artery, what of course will be the waste, the blood that is rich with waste, and we exchange that for oxygen-rich blood to be returned in the umbilical vein through the circulation of the chorion. So that's the focus of these very early steps. We have to establish that type of a circulation to bring in good blood from the placenta and to flush out and deliver that to the cells in the embryo and outside the embryo, and then get the waste back to the placenta so they can be exchanged to the maternal blood, and we have a real good situation. And of course, the maternal blood will be responsible for clearing all of those materials, those waste materials. So the heart and the great vessels form within the intraembryonic mesoderm in the cardiogenic area, and it begins as a pair of tubes that fuse to form a primitive heart. So there will be bilateral systems originally. So you picture two tubes side by side, and eventually they're going to bump together and just kind of open up and fuse and become a single linear tube. So the heart joins with the blood vessels of the chorion, so there's this growth in length, mitosis of cells along these columns. It bumps into other ones. They fuse, open their extracellular cavities, and the fluid within there, the plasma, eventually is just going to get pushed through everything. So eventually this heart's going to pump and put a pressure head on everything, so it's going to change the physical dynamics of it all very early on as some of the major connections between all these blood islands and early vessels will have been established. And so it'll get right through the connecting stalk to the extraembryonic vessels and through the vitelline artery and vein as well. So at the end of week 3, the blood is circulating and the heart is beating. So the beginning of week 3, we're just establishing the 3-cell layer. By the end of week 3, it's well beyond being established as a 3-layered embryonic disc. We actually have a circulatory system. So this would be the appearance of a 16-day embryo from the lateral view. So this is 2 days into week 2. So we can see that there's quite a good development of all the structures. We already have a heart that has migrated from the front of the head down in closer to where the chest cavity would be. The yolk sac is telling us where the intestinal structures are going to develop. And, of course, we see the somites along the edges, and the embryo is contained within the amniotic sac. So we see this then as the first organ system to develop where it is functional. So the other organ systems have primordia. The nervous system is developing quite rapidly, but it's not really functional yet. It's establishing, making touch with things, establishing contact, but the circulatory system's got to get fired up first. So it's detectable. Of course, this means from outside the uterus by week 5, so listening, auscultating the abdomen of the pregnant female or using ultrasound, it can be detected. So the blood vessels extend into the tertiary chorionic villi. We saw that. The fetal blood, maternal blood supplies are separate, but they can exchange the gases across the membranes at the villi, so we'll take a peek at that. So days 18 to 21, we see there's quite a few other changes, so many more somites by this stage. The yolk stalk is well established. The heart is now beginning to fold, so this long tube doesn't have space anymore, so it's kind of getting scrunched, and it'll do a little twist. I don't know if you've ever taken a hose and you twist it, and it'll kind of twist and then bend down and fall into another plane as you twist it. That's kind of what's happening here. It's one way of thinking about it. We see tremendous changes where the arteries are passing through the mesoderm of what's the face and the neck region, these arches, visceral arches. The nervous system is undergoing a great deal of development, develops more rapidly. In general, everything develops more rapidly in the cephalad or head region than it does in the caudad or the tail region. So we see the yolk stalk and the body stalk. They're starting to get into a location where we know the umbilicus is going to be formed. So that's day 18 to 21. Now we can look at figure 414 to see diagrams illustrating the secondary chorionic villi and begin to analyze what's happening there. So we see in panel B, there's a cross-section now. When we looked at these before as primary ooga-boogas, chorionic villi, got it out there, there was only the 2 layers. There was a solid core. We likened it to pushing our finger into some putty. Now we see that it's not a solid core, there is mesenchyme that has pushed up into the core of these secondary chorionic villi. And that mesenchyme, we can see closer to the extraembryonic coelom, and its margin there, the extraembryonic mesenchyme within the chorion is beginning to form the blood islands and blood vessels out there, and very soon these, of course, are going to begin to appear within this mesenchymal core of the secondary chorionic villi and form the tertiary chorionic villi that now have the vasculature. But what we see is that the maternal blood is going to be exposed to the outside of the syncytiotrophoblaст layer. So there's still quite a thick barrier between the maternal blood and the embryonic blood within these capillaries or blood islands. We actually have illustrated here a very complete circulatory system as we had shown in the previous slide. So we have the embryonic blood circulating now, and there is exchange, but it's not very efficient. So obviously the next transitions we want to make is to thin that out so that there is a very thin layer for exchange of nutrients and gases and waste between the embryonic and the maternal blood. So let's list and characterize the different developmental stages of these placental or chorionic villi. The primary villi have the cyto and syncytiotrophoblaст cells. The cytotrophoblast cell core is solid. Secondary, the cyto and syncytiotrophoblaст plus some of the extraembryonic mesoderm has pushed into there, into the center of the villi, and still it is a solid core. And then the early tertiary, there will be blood vessels in the mesoderm layer and the layers of the secondary villi. And the late tertiary villi, the cytotrophoblast, will be partially disappearing, and the syncytium and the endothelial wall of the blood vessels may be in direct contact by that point, and then the villi form richly arborized tree-like structures to increase the surface area exposed to the maternal blood in the maternal lakes, in the blood lakes. So we have blood islands on the embryonic side and blood lakes on the maternal side. The blood islands eventually all fuse together to form the vasculature of the embryonic cardiovascular system, and we see changes in the villi such that we thin out that membrane, separating the embryonic blood from the maternal blood. So which of the following does not contribute to the nutrition of the developing embryo? The amnion would be the correct answer to that. So the yolk sac, the chorion, and the decidual cells all make a contribution to the nutrition of the developing embryo from one stage to the next. So that completes Lecture 8, and I just closed all of them. Okay, so let's go to Lecture 9. So we've been discussing this within our discussions. We've constantly continued to mention high mitotic activity, high metabolic rate, so a tremendous consumption of energy for a teeny ball of cells, right to this sophisticated structure that has the chorionic villi and the body stalk and a complete circulatory system. So it's very rapidly growing, so it establishes its own circulatory system and connects with the maternal side of the circulation. Because it's growing so rapidly, and originally it has that purse string, the connection of the yolk sac and the amniotic sac, where the embryonic disc is located, we know that there's going to be too little room for things to grow as the cells are pushing out in a flat plane. So there's going to have to be bulging and folding and curving and curling. And eventually the flat disc is going to have to curl on itself to close up around the umbilical cord to form our belly button. So we know that has to happen. So now we're going to begin Lecture 9 talking about the folding in conjunction with reflections on the embryonic development at the time. So this slide is a little bit tricky when it first hits you, but we can get oriented quickly. We see the yolk sac with the developing blood islands in the splanchnic extraembryonic mesoderm. It's lined by the enteroderm. And then as we look, we see the cut edge of the amnion with the somatic mesoderm lining it. And immediately then when we see this dorsally placed neural fold, complex of the developing nervous system, the neural folds, neural groove, and the neuroenteric canal, we immediately realize this is a lateral view. And we can see the umbilical vein, the umbilical vessels. I screwed up there, didn't I? What's the blue vessel in the umbilical cord or the body stalk? The blue vessel is not the umbilical vein because it is blue because it's carrying the oxygen-poor blood away from the embryo heart. Away from the heart is artery, so this blue one has to be umbilical artery, taking it out for the exchange, and then coming back through the umbilical vein would be red. So I caught myself in one of my own tricks. We also see the allantoic stalk out here, so there is that early development of the allantoic stalk. So the very early umbilical cord is here at the body stalk. So this shows us the establishment of that circulation. And now the embryo is still constrained by the junction of the amniotic sac and the yolk sac is going to begin to try to stretch out in every direction radially. It's going to have to fold at all circumference. So that ultimately it collapses on itself, if you want to think of it that way, and brings the head region in, the tail region in, and each side in, so that it encircles the body stalk and forms the umbilicus. So we see that body stalk right over here. And then eventually the yolk sac will be contained within the umbilical cord as well. So tremendous mitotic division. Along with this, morphogenesis, changes in shape. So the shape of the embryo is going to change tremendously. So there's elaborate cell movements to form the primordial tissues and organs. All of these dynamic forces are acting within that purse string constraint. And so the 4th to the 8th week of development can most accurately be referred to as the organogenic phase. So by the time we finish week 3, we've got a circulatory system, and we're developing a lot of the organ systems. So this is the time when we are generating the organs. So the organogenic period is what we are talking about. So we also are thinking of the differentiation. So we have the formation of specialized cells and organs. So now we are thinking in our mind cytogenesis, histogenesis. We're developing different tissues, epithelial tissues, connective tissues, and things like that, and muscular tissues. If we're going to have a pumping heart, we're developing muscular tissues. So histogenesis and organogenesis and organ system development generation as well. So the organogenesis is what is occurring now, week 4 to 8. After 8 weeks, we just develop or finalize the development of these established organ systems so that they are prepared to present for meeting the needs of the newborn. So now we're thinking of this term induction. It's that complex interaction among the embryonic tissues to convert the pluripotent cells into dedicated cells. So this is nothing new to us. We're talking about it all along the way. So there are induction signals. Most of these are signals between cells, intercellular, not intra, inside, inter, between. So this means that they are extracellular. Many of them are even matrix proteins in the extracellular connective tissue, if you want to think of it that way. Extracellular matrix is this connective tissue that we think about. So there will be actual cell-to-cell physical contact, so there can be even direct intercellular communication. So not just by squirting chemicals at each other that diffuse out and might be delivered to the circulatory system, they also will begin to communicate as soon as they make physical contact. So let's look at an example of this based upon the neural crest cells. So we're seeing the early migration of the trunk, meaning in the trunk region, the thoracoabdominal region, the neural crest cells. So the yellow coloration is the epithelial, the surface ectoderm, just at the edge of the junction with the neural plates. This is where there's active mitosis with some stage of differentiation to produce the neural crest cells. Over here on the right, we see two panels of actual scanning electron micrographs of a mouse embryo, and we see the notochord down below here, so we know that's ventral. Dorsally is the surface ectoderm up here. This is the neural tube dorsally, and there's a little cluster of cells just dorsolateral to the neural tube near the somite of this developing from the paraxial mesoderm here. Next to that, they are colored red, and we see that there's a long trail of them. They are truly migratory, and they will move to different locations, as we saw indicated in the last lecture. Some will migrate and sustain that dorsolateral position and differentiate into the sensory ganglia, the spinal and cranial sensory ganglia. Some will migrate farther away, like out around here to the front, in front of the notochord. The aorta is going to end up there. So that's where a lot of the peripheral sympathetic ganglia are going to develop. Some are going to migrate into the head region to get near the salivary glands. So they migrate in various directions and various distances. If this is the upper abdominal region, some of these little buggers are going to migrate out to the intermediate intraembryonic mesoderm and differentiate and join with the mesodermal cells that are going to form the adrenal cortex that will encase these migrating neural crest cells, and the neural crest cells will differentiate into the adrenal medulla. We focus on the production of sympathetic neurons and adrenal medullary chromaffin cells in this lower panel as an example of this lineage of differentiation. So we have an example of the regulation of the terminal differentiation of truncal neural crest cells shown here. So there is a pluripotent, so it's not totipotent. It can't become anything and everything. It is pluripotent because it can become more than one thing. And eventually, through some mechanism, it proceeds to a point that it is now only two possibilities. It is a bipotent precursor cell. And depending upon its location, it will be exposed to different extracellular molecules. If it is close to the developing adrenal cortex cells in the abdominal region, they will be exposed to the steroids produced by the adrenal cortex. The glucocorticoids is one of those types. when when one of these bipotent precursor cells is exposed to the glucocorticoids, it will go through a next stage of differentiation to become a chromaffin precursor cell, and as the presence of glucocorticoids persists at a really good solid concentration closest to the source, it'll go through final differentiation to become these secretory endocrine cells, the chromaffin cells. We know that they secrete adrenaline and noradrenaline, and they also secrete some peptides. So therefore, we see these large secretory granules in the cytoplasm. Cells that migrate to different locations near the aorta predominantly, and of course all along the vertebra to form the paravertebral sympathetic chain ganglia. These cells will be exposed to fibroblast growth factor 2, so FGF2, and fibroblasts are connective tissue cells that differentiate from intraembryonic mesoderm. So these FGF2 cell exposed cells now will change their gene expression in a way different than these exposed to the glucocorticoid cells. They're going to display a lot of different genes such that this cell will now become nerve growth factor competent. So one gene it will express is the nerve growth factor receptor gene, so that the nerve growth factor receptor proteins will be present on the surface of this cell. Nerve growth factor will be secreted by nearby cells and bind to the nerve growth factor receptor on these precursor cells, the competent cells, and signal to them to make their final stage of differentiation to produce the axon and the dendrites and differentiate so that it expresses the genes for the synthesis of the molecules it's going to secrete, all of the enzyme in the biosynthetic pathway for the catecholamines to make adrenaline. And they will differentiate into actual sympathetic neurons. It is possible in adult adrenal, adult adrenal medullary cells to expose them, to dissect them out of the adrenal medulla and to put them in culture, and if you treat them just right and expose them to the appropriate factors, they will differentiate into sympathetic neurons. So it's possible to take these cells and produce these cells. So here's another view of this, the cross-section of the embryo with the neural tube shown here dorsally, a somatic mesoderm, mesoderm shown here, and the migratory pathway of the neural crest cells. So some moving down here in front of the notochord where the aorta is going to develop, others out to where the adrenal medulla might develop, and others out into regions where they might form parasympathetic ganglia. In addition, of course, their processes, the peripheral processes, will be extended out with the structures that they made contact with very early on. So on this panel here, we give an example to emphasize the changes, the progressive changes in gene expression as many of these events occur, when we have migration and then we have them becoming stationary and undergo their final terminal differentiation. I will not ask you to know these details, but they should be familiar enough to us. First, I'm remembering it, the FN stands for fibronectin, the LN stands for laminin, and the NCAM stands for neuronal cellular adhesion molecule. So it will make neurons stick to each other. So when this expression, the green, is high, the cells are going to stick to each other. When that expression of that gene and the presence thereby of the NCAM proteins on the surface of these cells is low, these cells don't stick to each other and they can move. So when NCAM levels are high, protein and gene expression, as the cells will stay clustered and clumped together as they are here early on near the neural folds. Something tells those genes to stop being expressed, and then the extracellular matrix cells, the mesodermal cells, are going to increase their expression of genes and secrete fibronectin and laminin proteins into the extracellular connective tissue matrix, and this provides the surface, the medium, for the neural crest cells to connect with so that they can migrate to their destination. Once they reach their destination, they've arrived to the site of differentiation, then the extracellular matrix will no longer contain the high levels of those migratory roadways, and the cells will express the molecules that make them stick to each other so that they stay where they're supposed to be and can undergo their final differentiation to begin their functional lifespan. So morphogenesis is a term related to variations in gene product, effects on cell shape, altered gene expression due to cell differentiation, cell shape changes that alter gene expression, collective alterations in groups of cells, embryonic folding. Of these, we're going to think of shape. Morpho is shape, so we have shape here and we have shape here. Shape does not drive gene expression. Gene expression, to have a product, a protein is a gene product, that is what is going to bring about, effect means to bring about, the cell shape changes. So this first response would be the correct response based upon that line of reasoning. Differentiation of neural crest cells is most affected by, so the differentiation of neural crest cells. Fibronectin is going to affect migration. Neural cell adhesion molecule is going to affect, is the term I'm using here, is going to affect the sticking of the cells together. That's not really differentiation. Most directly, now that we're getting the best possible answer. These are all involved with these cells undergoing a differentiation process, part of which is migratory or migration or unclumping, migrating, and clumping, but we're really thinking about the most direct effect on differentiation itself. Extracellular matrix, no. Cell membrane protein gene expression, not so much. Glucocorticoids, yes. We saw the glucocorticoids have the direct impact on taking a bipotent cell and telling it, you are going to become a, an adrenal medullary cell. And remember, this occurs in the presence of glucocorticoids. The glucocorticoids are present once the cell has reached its site of differentiation. So once these neural crest cells undergo all this change in gene expression of NCAMs and interfaces with the extracellular matrix, gets to where it's supposed to be, and then sticks together, that's where it's finally exposed to the highest levels of glucocorticoid that signal it to undergo its final differentiation. So that's why this is the best possible answer to that question. So now we can begin to look at the embryonic folding. So it occurs both in the horizontal and the median plane. So horizontally and through the in the median plane, we're going to see certain foldings. And this rapid growth, especially the nervous system. The nervous system cells of the neural plate fold and ultimately tube is just going to be very rapid and push and swell forward and form these vesicles. The lumen of the neural tube is going to fill with fluid in the front part, the cephalic end, or the cephod, and it's going to swell, so it's going to push things forward. Something has to happen. There has to be a displacement. So this brings us to this question. Agents as drugs, such as drugs, and viruses that produce or raise the incidence of congenital anomalies are called what? Teratogens. So these types of molecules that produce congenital anomalies are teratogens. They alter the production of or let's say the orchestration of morphogenesis. So you get morphological anomalies. And it's important to bring it up here because we're in week 3 and week 4, and an individual may just, let's try to add it up here. We've got day 14. At the end of 2 weeks, an individual would expect the beginning of their next menstrual flow. So week 3 and week 4 are the weeks where they're questioning, am I really missing my period? Am I really pregnant? And so it's still quite early on, so there might be exposures to substances up to that point, and it could have tremendous effects. So the later in the pregnancy that things happen, the less likely they are to impact morphogenesis. So let's take a quick look. We recognize this dorsal view. The cardiogenic region is here. The oropharyngeal membrane is here. The neural fold is there. We're going to begin to see a head fold, a tail fold, but then a cross section. We take a look at it. We're going to begin to see these tremendous lateral folds. So in the median plane, the cranial fold, about week 4. So this is the forebrain and oropharyngeal part of the, that's going to be developing rapidly and get pushed forward and only have the choice to curl under. The heart's there as well. It's going to move into the ventral aspect of the embryo, and the yolk sac and endoderm becomes the foregut, the esophagus, and the pharynx as it's drawn into this head curl. Let's see how this looks then. The heart's coming down, oropharyngeal membrane's dropping around to the front of the head region, and the brain's pushing up into the top of it. So this little bit of yolk sac that gets drawn up in there is going to become the foregut. And then we're going to see a tail fold back here, bring the anus into place, and the yolk sac drawn in is going to be the hindgut. Everything in between will be the midgut. Not such of a concern right now, but it will be later. And then we're starting to close around in on the sides to close off the umbilical cord. So that caudal fold is in the median plane as well, and we're getting the distal tube. The cloaca comes around to start making the bladder and the rectum. We get the hindgut, and then the connecting stalk is going to become the umbilical cord as these tail fold and head folds continue to migrate toward each other, toward the midline, the belly button. But as well, on each side, we've got the lateral folding occurring to bring everything together. So we picture ourselves all stretched out, and we've got to bring our heart in front of us and our buttocks in from behind us. We've also got to bring our sides in to come around and surround the umbilical cord. So that horizontal plane, we're getting the right and the left lateral folds, a lot of growth of the neural tube. Everything's just kind of pinching together. There's no place for things to go except to constrict down upon it. And, of course, the endoderm forms the midgut, as we had seen. So this region here is going to be midgut. This back here is hindgut, and that's foregut being drawn way in. Heart now is drawn very tightly into the chest region. And we see the intestine is being surrounded by mesenchyme, and the lateral body fold is approximating anteriorly to close off the anterior body wall. So we can do this quickly in just the last couple of seconds here. High mag of the rostral end. This is growing fast pushing up, so there's no place for this to go but to push around in. We want to bring the heart into the chest region, the oropharyngeal membrane into the area where the heart would be. So we see that pull around, so the mouth faces the front and the heart is going to be in the chest. The pericardial coelom, we know that this is continuous with the pleural and peritoneal coeloms as it formed that original horse-shaped structure. The heart's developing in here. So after folding, the pericardial coelom lies blank to the heart and blank to the septum transversum. So the septum transversum is forming back here. So the heart is going to be rostral to that, and the pericardial coelom is going to be ventral to the heart. So the pericardial coelom lies ventral to the heart and in front of, cranial or rostral to, the septum transversum. The caudal end is going to be experiencing the same type of a situation, and we see everything coming in and closing around to form the umbilical cord in that area. So if we took a cross-section of the embryo at this time and looked at it three-dimensionally, again, this is a lot of information, I know, but you don't need to be an expert at it yet. We're just seeing where it would occur. So we see the pleural pericardial peritoneal cavity is here. It's all continuous. The heart is here, pulling down into where the lungs are forming. So everything's getting where it's supposed to be. So I will leave this question to you and any additional questions that might show up. And again, of course, we finish with the review of the germ layer derivatives in these lists. We have talked about them as we have been progressing. So it basically is review. So I appreciate your time and attention. We'll see you in class on Monday. For those of you that want to stay, I know I did say that I would show you the slides as they were presented for the first exam. I'll just get the CD in here. I have found that we are going to be able to load them, so it's going to work well. So I'm not concerned about that. But I do want to, if you want to sit and just watch for a minute, I want to show you that mechanism that we used previously, and hopefully you'll end up liking the way that it is done now. So this is how we had it presented before. These were the figures. You're in class with the written exam in front of you, and I gave 1 minute for everyone to answer this question. Here's this slide. You see this area here, this type of tissue on each of these adjacent structures. Different types of cells in here. They change their appearance as you go from this structure to this area. So what is this structure overall, and what process occurs here? So you have the choices will be there. So this will show up, it'll pop up with the question on the online exam. These questions will be there, and the choices for answers will be there as well. So you will just choose one of the answers that you feel is correct. Here we see this structure. You see this structure here, and these here. This has a question mark. What is this structure with the question mark on it? And what cell type is found out here? So we covered this in lecture. Exact same slide, basically the same information presented then. The highlighted structure here in pink appears between. So what is it between? It's between these things and this thing. So what are they? The highlighted cells originate from what? So it's the cumulus ooropharus, and of course they arise from the granulosa cells. The highlighted cells are, of course, the ciliated simple columnar epithelium of the uterine tube. what are the dilated structures in the boxes? We did this in class. Those are the uterine glands. So there are those slides that will be there. They will be presented without my oration to introduce them. Shouldn't be a problem for you, though. And in addition, there are, I think, 3 or 4 illustrations. There are numbered illustrations where we labeled each number in class. So a few of those will show up. And then there will be the typical multiple choice types of questions covering the lecture material in lectures 1 through 10. So everyone have a nice weekend. If there are issues that have disturbed you with us not covering all the lecture material, share those concerns with me so I get a feel for your, the degree of anticipation and anxiety that you're getting, and then we'll see how the exam turns out. It may be that you're getting nervous and upset and you get 100 on the exam, and for me, that'll let me know in the future that I can very readily comfort people. And, of course, in the future, we'll have everything pre-taped, and it'll be on there. So I appreciate your efforts and continued interest in the class. We'll see you on Monday. Have a good weekend.